Okay, guys, good evening. Yes, so we are back again. So, module 3, talking about databases, both for macromolecules, proteins, DNA, lipids, small molecules, chemical libraries, and all sort of that. So, that's what we are discussing in this practical section. And as I mentioned in the course tutorial, I'm just going to walk you through so many of them to give you a feel of what and what you need to do around them and then how to use each of them. Yeah, because each of them have a specific usage, right? Okay. Yeah, so we'll start from PDB, the Protein Data Bank. This is the site and it's maintained by all these hosts. Um, yeah, so PDBs are a repository for proteins, DNA, antibodies, macromolecular structures, certainly. So either from X-ray crystallography, NMR spectroscopy, cryo-electron microscopy, or whatever have you. So as of today, we have 257,000 structures available, both as APO, that means no ligand bound, or as a holo, no cofactor bound, or as a complex bound to a ligand. Then also here you also have protein-protein complexes, not just protein ligand complexes. You also have protein antibody conjugates, DNA complexes, and on and on. So it's it's a comprehensive database, trust me. And it's a very rich resource for people in whether you're molecular docking, even if you're trying to build an algorithm, machine learning, and all sort of things, you need this database. So first, let's start. Um, each entry in PDB, Protein Data Bank, contains 4 letters. So we're going to use this 9NSR. So these are 4 letters, and if it's available, it will pop up. If it's not available, it will tell you. As you can see here, it's predicted that by 2028, the PDB will have exhausted all the possible 4 letter combinations that are available. So it's like 10, 9, 9 letters from the numbers 0 to 9, and then 26 letters. So all the possible combinations would have been exhausted. So by 2027, they are going to move into a 12-character PDB ID, and then even the format, which I'm going to show you very soon, will change. It will no longer be a PDB format, but it will become PDBX or MMCIF format. Okay, so crystallographic format. So and all that. So yeah, so but for now, it's still 4 letter code. So when you come, you search, and then you see what you're looking for. So a whole lot of things are here. If you look at this bar here, you are going to see so many options are available to you. You want to see the structure, the annotations for people into structural biology, the experiments that is behind this. You want to see the sequence of this particular protein, the genome, ligands, or whatever is available. And this is just a small snapshot of the protein. You can also see some of the annotations, a structure, sequence, electron density, and everything. Now, most importantly is the okay, as I mentioned initially, the PDB not naming system, I mean, the PDB ID will change into a 12-character word. They have already started implementing it, but it will go fully live in 2028. So, as you can see, this 12-character word that is already encoded for this 9NSR. Now, something that is important, something that is important is the information that is here. I mean, the title, let's say that way. So usually the depositors will tell you, oh, this is an apo structure, or this is a protein that is co-crystallized with a ligand, or a protein that is maybe it's a complex, or it's even a protein conjugate or whatever. So you just have it mentioned here. Possibly they will even tell you it's from Plasmodium falciparum or is a human insulin or whatever have you. So that is important. So next is the classification, the enzyme classification, the organism where it's gotten from, and possibly the expression system. So as you know, usually the expression system is different from the organism where it's gotten from another. Right? Yeah. So now, something that is important here is the experimental snapshot. That is just a summary. So you're going to see, oh, it's coming from X-ray diffraction, or it's from NMR spectroscopy, or whatever. Then something else, the resolution. So the resolution tells you how accurate they are about the position of each of the residues or nucleotide in your macromolecule. So the lower the number in the resolution, the higher the resolution. So 3.25 resolution is a poorer structure compared to 2.0 Armstrong resolution, right? So R value free method, R value work, R value. So these are some parameters that were used during the fitting. If you're into crystallography, you understand more. But for now, they might not be very, very important to us. Now, when you go down, usually there should be a literature that is associated with every deposit. Literature here means paper. Someone must have published something associated with this kind of deposition of structure. But some other times it's not available. Like you see here, this one has not been published. It's possible it's a preprint already, maybe in a database somewhere, but they just decide to deposit it while they wait for the publication to happen, right? Okay, so now, in this section, we are going to see all the content of this protein. So we're talking about the chains. For example, here, this contains only one chain made up of 378 residues, right? And then if it has a ligand, you are going to see that. So this is a ligand that is co-crystallized with it. Now, some other times, this is just a very interesting protein because it doesn't have so many issues. Now, let me show you some another one that might be also interesting. So this guy is dihydrofolate reductase thymidylase synthase involved in folic acid. And so as you can see, this guy's already published a work associated with it. Importantly, you can see so many chains, chain A, chain B, each of them 280. There's a chain C, so there are 4 chains available in this protein. Now there are cofactors, NDP, there's UMP, then there's co-crystallized ligand, and on and on and on. So all these things are important for you to evaluate your structure. Now, if you want to choose a protein for molecular modeling or docking, the important things are, okay, there's something I forgot. Something that has to do with mutation. Is mutation available in this protein? The answer is no. So you have to check, oh, do I have mutations? Because if there are missing residues or there are mutations or some places are not very clear. For example, here there are no mutations, but if you look at the structure, you are going to see that there are some missing residues, as I'm going to show you now. And then if there are those missing residues, it simply means that we need extra layer of modeling. As you can see here, clearly, you can see missing residues already, right? So, yeah. So now, if these missing residues are available, so it means that, oh, you need extra level of, let's say, filling the missing residue, filling of loops and all that to be able to make the structure very, very attractive for your use, right? Okay, but essentially, once you check the resolution and the mutation, then you are just very fine. So something important again is downloading the data. So if you click here, you can get the faster sequence for this particular protein. Yes. So, if you want to work on modeling, homology modeling, or you want to use AlphaFold to do modeling, you can find faster sequence for this file. You can download it. And as I mentioned, because some of these structures were deposited prior to now, they still have the normal PDB format, but very soon everything will become PDBX, the MMCI format. So it's just an extra encoding that will allow more representation of all the structures available in the PDB protein data bank. Right, so you have a lot of things that you can download. So it depends on what you are doing. If you're just trying to do docking, you might just stay in the PDB. If you are really into crystallography, there are many other things that might interest you. So that is actually what to do. Now the most interesting thing about PDB is that it has application program interface. So that means sets of codes that can allow you extract or retrieve these structures without coming to the graphical interface this way. Or let's even say you have a lot of structures. Let's say you want to retrieve all structures that are available all proteins available for Plasmodium falciparum from the PDB database. So they allow you provide all manner of IDs, supply them, and they are going to retrieve them for you. So if you're working on Python, or you are doing throughput, you are doing machine learning, you are trying to build an interface for, an algorithm maybe for docking, trying to optimize force fields or whatever, you might want to use API, because by that time you may request, oh, I need 300 proteins from the protein data bank. Bro, you can't download them singly, right? So you might want to, you start trying to move around how to use these APIs. And the funny thing is PDB provides tutorials. I would have shown you this maybe earlier on before now. Yeah. So if you check PDB 101, you are going to see so many tutorials that are providing for people to learn how to use PDB, everything about PDB. So if you're interested in structural biology, interested in whatever, you might want to actually stay around here for some time. That is interesting. Okay. Now, they even organize courses, as you can see, learn, teach, whatever. So they organize a whole lot of things. So now, let's talk about some other database. The next one is Uniprot. So Uniprot is actually, it's the database for genes, let's just say at the genomic level, right? So it's not just about structure, just like PDB. So PDB is just dealing with structure, but Uniprot is about genomes, about genes, about structure on its own, about everything that has to do at the genetic level. So it's not chemistry related, it's just biology, structural biology. So here you are going to find enzyme classification, genomic variation, single nucleotide polymorphism, all manner of things. So, and usually you start by putting in Uniprot ID. So for every protein, whatever, there is an available Uniprot ID. And then if you insert it, you're going to find the protein or the gene associated with that. So for example, this is lysosome C enzyme or protein. And then you can have a lot of things about the function, catalytic activity, even evolutionary tree. Just so structural biology. So, yeah. And then you can look at so many things. Where does it belong in enzyme classification and all sorts of things? Where does it localize in the cell, proteomes, phenotypes and variants. So it's a whole lot of things. So if you're working at molecular biology or at the level of genomics, you might really want to hear you're studying variation, variants, even precision medicine, all that. So I want to admit I'm not an expert. I only use this sparingly, but yeah, it also reflects on the kind of things I do. So I'm more chemistry inclined. So I just come here once in a while to pick what I want. And so something that is important is that it's going to provide you the structure of the protein. It's either it's available in crystallography or it has been predicted by AlphaFold. So for example, for this particular enzyme, A lot of people have worked on it. There's so much crystallography data around it. So that's actually, for example, you can see some of them were predicted by AlphaFold. Now, that's actually very important because sometimes I'm looking for a protein and I want to just quickly see how many PDBs are available for that particular protein. So I'll just come here and look at them and even see the different resolutions. So you get, so it's something that can be very, very quick and also fast. Then if you're also looking for, let's say, the sequence, maybe you want to do modeling for AlphaFold and just looking for the primary sequence is available here. You can also pick it up. And a whole lot of other things that um I mean, sorry, that are available here. So I just want you to know that this exists and this will literally change how you do your maybe computational biology work or what have you. Okay, now moving on to PubChem. So we are now moving from generally protein to, let's say, um, small molecules, right? So we're talking about natural products, synthetic products, assays, and all that. So PubChem is a repository of small molecules, so compounds. So you see, you can search by SMILE notations, the name, you can even search by organisms. Usually when you search by organism, it retrieves assays and compounds associated with them. If you just search by a compound name, for example, aspirin, then, and then there are so many variants. I mean, structural modifications available. For example, aspirin, dilysine, sorry, D-lysine, that is a racemate, aspirin and hydra, whatever have you. So if you're just looking for plain aspirin, it's also available. So once it presents the structure, then you can go in and then look at the structure. So this is actually where you retrieve your compounds for molecular modeling studies. So the 2D structure of your compound, the 3D structure of your compound, and then even crystallographic structure, everything is available here. And all the information about your compound. So look at the structure, and okay, if you're interested in chemical feeling, whatever, then you see all the names, all the chemical identification system available, IUPAC name, INCHI, SMICE notation. So SMICE notation is also available here and every other thing. Now, something that is very, very interesting is usually, sometimes it's not even the physical properties and all that, but something that is interesting is usually assays associated with aspirin. So you to be provided and you can see, oh, aspirin has been studied maybe as an anti-cancer agent using this cell line, and then maybe the IC50 is this, because those things are important when you start building machine learning model, because you will retrieve this information and then use them to build your models. So these informations are usually available lower down here. Let me stop scrolling. And meanwhile, there are a lot of things here like pharmacology, manufacturing, whatever have even patents associated with aspirin. For example, you can see a lot of patents associated with aspirin. Not necessarily that many people patented the same compound, but it's just patents associated with maybe a use case or a derivative of aspirin and all that, right? Now, important is biological test. That's actually where I was going. So you can see a lot of bioassays. You can see 7,000 possible bioassays associated with aspirin. So has been done. So these are important quantitative and assay data that are used for molecular modeling or the building machine learning models and all that. You get. So that's actually very important. And usually you don't retrieve them one by one this way because it's crazy, bro. You are not, your name is not Job, right? Okay, so usually we use APIs to retrieve them. I'm going to show you that in a code. So this is what actually happens in PubChem. Well, sometimes you might just be interested in getting, three structures. I want to just have these three structures. I want to use them for molecular docking. I want to use them as starting structures and optimize them for molecular dynamics. I want to use them for DFT, whatever is available to you. Now, you can click on downloads, and then depending on what's available, you can see the 2D structure, the 3D structure, or whatever. So usually you just download the SDF format, Yeah, the structured data format, and then you can do a lot of conversion with this cost, open Babel, RDKit, what have you. You can now do a lot of conversion. So that's just simple, something about PubChem. There's still a lot of things about PubChem, but I think most of them will come with use. Yes. So as you use them more and more, you will get to, sorry about it, you get to know about it. You can also cite the work if you're interested. If you use PubChem, you can have a lot of citations available for you. Now, Cambly database is also an analog, Pop came from the US, Cambly from the UK. So it has 2.9 million compounds. It has 2.0 million assays. So this is exactly just like PubChem. It's just like PubChem. It's a representative of all chemical compounds available, their assays, their properties, and everything, including their targets. For example, was this assayed against Plasmodium falciparum, or was it done against the Ratus, whatever have you? So, the same way you can search aspirin by name, and then you can retrieve chemical compounds, right? So you can see aspirin and so many of the derivatives. It's just similar to uh, PubChem, right? So you can see a lot of things, structural alert, literature, activity charts, so many things are available here, presentations. So as I mentioned earlier on, you don't download them one by one, especially if you have a lot of compounds to work with. You need 10,000 compounds. That's not possible. You make use of APIs to retrieve these compounds. Okay, let's just move on. Yeah. Now, drug bank is for drug repurposing, right? So if you're a student and you're interested in seeing how can approved drugs be utilized as starting points and not necessarily as endpoints, but maybe starting point for further modeling, optimization, or even, um, I mean, test them on newer targets or orthogonal, whatever it's your goal. Yeah, I mean, whatever is your goal, right? Okay, so you come to a drug bank, it provides all these things. For example, you can also search for aspirin, and then it's going to show you what compound is that, and all that and all that. Now, usually I've used drug bank before, so usually you create an account and then request for all the available drugs here, and then they'll give it to you under academic research, right? And then you can make use of them. So usually they give it to you as a single SDF file. I said I don't know why they don't believe I'm a human being. It's fine. It's their laws. I'm a human being. Okay, so usually come as a single SDF file that you can use. So I think we've discussed that on OpenBB. You can split the compounds. They're asking me to create an account or log in, whatever I want to do. So, but these things are available, right? For academic research, these things are available. You can ask for it and they will send it to your mail as SDF file. Then you can just split them out, retrieve all the compounds, and start using them for whatever modeling you're interested in. Yeah. Okay. Now, what of natural products? What if you're interested in natural products? What do we do? So coconut database is the largest collection of data natural compounds available. I think as of today, we have like up to, okay, you have like 738,000 compounds available in coconut, right? So I think it's even more than that. Yeah. So it comes from a whole lot of, I mean, a whole lot of regional databases. What I mean is compounds that are peculiar to West Africa, compounds that are peculiar to the South American continents, and whatever. So this is a serious and thorough compilation. Right? So you can search by compound name, you can search by SMILE notation. Katekin, I'm not sure whether I spelled it right, but I'm going to try. So and then these compounds will be retrieved. Okay, I'm not sure whether I spelled it right. Okay, taking. Okay, so I now got it. So you see, catechin and all possible derivatives of it are available, provided for you. And then you can actually, you know, download. So there's a download option. You can download each of them separately, but usually you don't work on each of them separately. Usually when you want to apply this for, a screening workflow or whatever, so you go to the download section and then they're going to, just like I told you, it's an SDF file, and then you split them, right? So just again, multiple files concatenated together, so you can just strip them and start using them. So if you want them CSV format, so usually they will supply the smiles for each entry. If you want them in SDF, you see it generated is using RD kit. So if you want them maybe separated, active, inactive for a particular one, so you can get all manner of things here. So also, if you want to search the database, it's also possible to search it by making an entry from a structure. So you can add a structure and then let's say what have you. Okay, I think I need this. Yes. So let's just have a nitrogen here to help us. So if I have this substructure, I can say, oh, I want to do a substructure search, right? Yes, so, or maybe an exact match, or let's just stay with substructure search, and I want to search for database. So you can use that. So if you have a particular scaffold that you want to select similar scaffold from this database, so they are giving us up to 35,000 outputs, and we can actually download them, right? And then use them for our screening. So these are all natural products, alkaloids, whatever have you, they're available. So you can do some of this substructure search, and that is actually very interesting. So if you want to see various collection, as I told you, it's a comprehensive database, including regional specific databases. So you can see natural product from Costa Rica, the South American continent, and whatever have you. I think this is from Peru and all manner of things. So I think this is even from European Union and all that. So Australian. So if you want to be specific to each of these regional databases, you can just be there. So I've used this and it's really very interesting. I'm still planning to use it again. So please come around and use it. Yeah, the authors have done a good job on it. Now, moving on, we mentioned chemical libraries. So these are chemical libraries on their own are not just databases of known or available compounds or even tested compounds. Sometimes they are database of hypothetical compounds. In fact, they are called synthesis on demand. That means you can come there and say, oh, I like this molecule. Or you collect some compounds from there and then they are hypothetical. They were generated using a whole lot of things, combinatorial chemistry, click reactions, whatever. Then you say, oh, I dock these compounds. Please, can you send them to my lab? They'll say, oh, yeah. They will synthesize them. So they don't really synthesize these compounds and keep them for you. So they are virtual, exactly. That's the right word. They are virtual databases. So an example is Enamine. So Enamine is one of the largest collection of databases. I'm talking about billions and billions of compounds that is available in their database. You get? So it's a custom synthesis. So when you demand, they are going to give it to you, right? When you demand, they are going to give it to you. So now I've used enamine and I'm going to just show you maybe, one or two ways on how to go about it, right? Okay, so you can come to compound libraries and you say, oh, I want to work on bioactive libraries. That's libraries of compounds that have been proven to be active. Okay, see, for FDA approved drug, they even have a list similar to drug bank, although it is less in number because it contains everything from the WHO drug list, so you can easily download it. Or they say PAINS library, the pan-assay interference compounds that are frequent hitters when you do molecular docking or assays, so you want to collect them or whatever. So you can go by that. You might be interested in, oh, I want to work on targeted libraries. I think we mentioned targeted libraries. Oh, libraries that are associated with allosteric kinase activities, or maybe you're working for anti-tuberculosis. So all these things are synthetic compounds, but you might want to use them as starting point for your virtual screening, right? Or maybe you're working on a viral compound. So instead of going to PubChem and collecting all manner of structures to start, you might come here and focus only. There are 3,200 compounds here that are known to have antiviral properties. So you start from there. After docking, you start modifying, optimizing them. Base 1 library for working on Alzheimer's diseases and a whole lot of them. GPCR, protein coupled receptor, G protein coupled receptors, if you're working on that. So, and a whole lot of that. So these are available here and all of them are free to be downloaded. As I told you, they just want you to work with them. If you now find something interesting, you can say, oh guys, I'm interested in this compound. Can you synthesize them for me? And they will say, oh yeah, that's our job. In fact, that's how we survive. They will send it to you. So that's exactly, this is a rundown through so many of the databases that I intended to show you. Well, this is short, and I know I was not very much in details, but my job is to make sure you are aware. And trust me, this is not exhaustive. There are still many, many available databases. Now, let me just show you how to retrieve compound using APIs, right? So this is a workup for module 3. Remember, we're in the practical section of module 3, target and compound databases. So we're going to retrieve some compound from PubChem. So usually to retrieve compounds, you can do a quick search for assay IDs. They are called AIDs, right? Associated, for example, here, we want to retrieve compounds associated with Plasmodium falciparum activity, right? Antiplasmodial active compounds, right? So our job is to find these assays because activities or testing activities of compound against Plasmodium, they're usually done in batches. It's not like, oh, when somebody tests, sometimes it's like that. You can scrape the literature and deposit what people tested individually. But usually there are people that their job is to do these assays and deposit, right? So it comes in batches, it comes in assay IDs. So usually it's a collection of compounds. So you identify the assay IDs associated with the particular activity or the organism you're interested in. And then, for example, here we are going to ask it to retrieve all the compounds associated with this assay ID from the PubChem URL. You see. So I'm going to run this code. Now it's done. So let's visualize. I also added some codes here that you might want to you might want to save them out, right? Okay, so as a CSV file. So, but let's visualize it. Let's just head the file. So you can see here a whole lot of, there are 38 columns. Remember, it's a head. So there are not only 5 rows, there are a lot of structures here. And you can see at least we have the smile notations here. We have a lot of entries, activities, course, and then so many entries are also not available. So now, so this is how to retrieve. You even have potency values. So you have activity classification and all that. So usually when you retrieve this way, you now start doing your filtering. You collect the smile notation, you collect the PubChem ID, and then on and on and on until you get what you want. So this is actually how to retrieve your compound. Now the same way with Chembl. So for Chembl, it's even better because I asked, collect only the Chembl ID, standard value of the assay, the canonical SMILE, the ligand efficiency, standard unit and standard type, and then collect only 2,000 of them compounds. So there are more than 2,000. So maybe I can just try to make it 4,000, and I'm going to run the code. So because I have time, I imported time, so it will be telling me what it's doing per time. So it's going to extract the first 1,000, the second 1,000, the third 1,000 until it extract the 4,000, and then we can visualize it and see. So this is how to retrieve compound. I think I know some of you already do this, but I just want to show you. It's not going to the website and downloading one by one. You just need to look for a code and optimize it, and then it will work for you. Okay. So I think we are done, and then we can head the data set, and then we can see this already. So it collects the Chembl ID, the standard values in nanomolar, and all of them are IC50 values. You see, very, very easy. For canonical SMILE, some of them do not have SMILE, so you always filter. If it's not if it's not available, they are not strings or they're not what you want to filter them out. So I just wanted to show you this, and this is actually how to retrieve your compass. So this Jupyter notebook is made available as part of the course file. All right, so this is where we are going to call it a day, and I hope you have learned a lot, starting from PDB to Uniprot and what have you. And there's something about this is the more you go, the more you use it, the more you understand which of the database is useful for what I'm doing, and then you use it and implement it. Right. Have a nice day. Bye.